Microscopy is experiencing a revolution. Developers around the world are working together to push microscope technology to the extremes. The Wyss Center is joining them. With our lightsheet technology, we integrate innovative tools to look at the fluorescent signal within whole organs. We do more than microscopy. So we first prepare the sample and then remove the lipids from the tissue to make it completely transparent. With lightsheet imaging, we can now optically slice samples with a sheet of light. Our lightsheet microscope acquires images of the sample, and we use specialized software to combine the images and build up a 3D picture. And we can even visualize the entire 3D sample in virtual reality.
My research is focused on spinal cord injury and finding treatment to improve recovery. A revolution came in neuroscience. Suddenly we can visualize the central nervous system transparently without cutting it. For us, it has been a game changer. So really, this microscope has completely changed the way we are now addressing anatomy, and been really for the very first time to be able to say clearly whether our treatment have worked or not. Now we have been able to show the entire spinal cord and really see these regenerating fibers going through the injury and beyond. And without any doubt, you can claim a real, true regeneration of the spinal cord.
In my lab, we're interested in how the brain generates behavior. For this reason, we look at the activity of single neurons, and we obviously would like to understand how the neurons talk to each other. And for that reason, we need tools to look who talks to who in the brain.
After having imaged the brain, we end up with a volumetric map of some specific connections. But then we need to make sense out of that. We need to get numbers, we need to understand. So this is where the AI will come into play and help us to do this kind of analysis. Our vision is also to develop these IT tools by setting up collaboration with researchers, specialists in this field.
So if the technique can accommodate even larger brains, such as ultimately a human brain, that would help us to draw parallels between anatomy in mice and men. And that is certainly one of the ultimate goals.